Hi, my name is Grant Kramer, and I am a professor emeritus at the University of Nevada, Reno. Welcome back to my plant physiology series. Today we're going to go through an extensive but exciting lecture on auxin, a growth hormone. Plant Physiology, Lecture 14, Auxin, the growth hormone. In today's lecture, we will learn about the first plant hormone discovered. It's a very important and interesting hormone that is involved in plant growth and development. There's a lot in this lecture, so without further ado, let's get started. But before we start, let's review from last lecture the general responses of hormones in plants. This will be the model for all the hormones discussed in the following lectures. The hormone is at steady state concentration in the plant and can change in response to environmental or genetic signals. The concentration can change as discussed in the previous lecture by biosynthesis, degradation, which is also called catabolism, transport, and conjugation. The hormone is transported to the site of action where it binds to a receptor. The receptor triggers a signal transduction pathway that ends in a response or action to that hormone. The response reaches some sort of threshold and can be modified by positive or negative feedback, altering the concentration of the hormone by transport, synthesis, catabolism, or conjugation, and altering the sensitivity to the hormone by altering the affinity or amount of the receptor or altering the sensitivity of the signal transduction pathway. Auxin means to increase or grow, from the Greek word auxin. Scientific discoveries do not come out of the blue. They are based on keen observations, in most cases by multiple individuals over time. The discovery of auxin is a good example of that. In 1880, Darwin and his son Francis published a book called The Power of Movements in Plants. In it, they describe an experiment they performed while studying phototropism, the growth towards light. They planted seeds in the dark, and when the seedlings emerged, they shined a light from one direction. The seedlings bent towards the light. If they cut off the tip of the coleoptile of the seedling, it did not bend towards the light, or if they covered the intact seedling, it did not bend towards the light. They concluded that a growth stimulus was produced in the tip of the coleoptile and transported down the coleoptile to the growing region of the coleoptile below the tip, stimulating the growth of the cells on the shaded side. Peter Boyce and Jensen expanded on this research, publishing a paper in 1913 describing this phenomenon in more detail. He inserted a mica sheet halfway through the coleoptile tip on the dark side of the coleoptile. The mica chip blocked the bending action. If he inserted the mica chip on the light side, then bending occurred in response to light. He also cut the tip off and placed a piece of gelatin between the tip and the coleoptile stump. The coleoptile continued to bend towards the light. He concluded that this growth stimulus was chemical in nature and passes through gelatin, but not through water-impermeable barriers such as mica. In 1919, Arpad Pal showed that the tip could be removed and placed on one edge of the coleoptile, causing that edge to grow. The edge without the tip did not grow. This effectively caused the coleoptile to bend without a light stimulus. Finally, in 1926, Fritz Wendt was credited with the discovery of auxin by performing the following experiments. He cut tips off of coleoptiles and placed them on a gelatin slab. He removed the tips and cut the slab into blocks. He then placed the blocks on the coleoptile edges and measured the angle of curvature after a period of time. He showed that there was increased curvature with increasing number of tips on the gelatin block. Thus, the growth stimulus diffused into the gelatin from the tip and the increased number of tips increased the concentration of the growth stimulus substance, now known as the plant hormone auxin. Before auxin could be chemically measured, auxin was measured by this assay known as a bioassay. The bottom right-hand graph depicts this assay with the curvature plotted against the IAA concentration. IAA is the main auxin in plants. Note, at highest concentrations, curvature is decreased from optimum. This is a real phenomenon. At high concentrations, auxin promotes the production of another hormone called ethylene, which inhibits growth. So plant responses are complex. I will discuss more on that later. We now know there is more than one auxin. They are indole-3 acetic acid, or IAA, 4-chloroindole-3 acetic acid, and indole-3 butyric acid. IAA is the primary auxin and is by far the most abundant and physiologically important form. In some species, 4-chloroindole-3 acetic acid and indole-3 butyric acid function as natural auxins. Because the auxin structure is relatively simple, scientists have been able to synthesize auxin-like molecules. Two of those seen here are used as herbicides. They act in a superauxin-like manner by causing uncontrolled growth in the plants, resulting in vascular disruption and death. Okay, now that we know what auxin is, let's examine how auxin concentrations are controlled in the plant. Let's start with auxin biosynthesis. Auxin is synthesized in meristems, young leaves, and developing fruit and seeds. There are two different pathways, the tryptophan-dependent biosynthesis pathway and the tryptophan-independent biosynthesis pathway. The precursors to these pathways are tryptophan and indole-3 glycerol phosphate. The tryptophan biosynthetic pathway is shown on the left. The branch point precursor for tryptophan-independent biosynthesis is indole-3 glycerol phosphate. Indole-3 pyruvic acid serves as an intermediate. The names of single gene mutants are placed in parentheses. These mutants have been cleverly used by plant biochemists and geneticists to dissect and identify the molecules in plant biochemical pathways. Auxin concentrations can be controlled by conjugation and catabolism. Most IAA in the plant is in a covalently bound form, which is inactive. This figure shows the structures and proposed metabolic pathways of these bound auxins. These are conjugated forms of auxins. Double arrows indicate reversible reactions or conjugation and deconjugation. Single arrows indicate irreversible pathways leading to degradation or catabolism of auxin. Now let's look at the fascinating topic of auxin transport. Hold on to your seat, it's quite complicated. Auxin transport is said to be polar. It moves downward from the tip where it is synthesized. This is known as basipedal, which means from tips towards the base, whether it is from the shoot or the root tips. Acropedal is away from the base, towards the tips in a reverse manner of basal pedal transport. This can be seen in Figure A. Figure B shows how this can be detected. It is a standard method for measuring polar auxin transport. The polarity of transport is independent of orientation with respect to gravity. A hypocutal section is removed and a donor agar block containing radio-labeled auxin is placed on the upper end of the hypocutal, and a receiver agar block is placed on the lower end. After some time, the receiver block is collected, and the radioactive auxin that was collected can be counted in a scintillation counter. This is a very sensitive measurement and can count very low amounts of radioactive substance. When the hypocutal is inverted so that the donor block is placed on the lower part of the hypocutal, no auxin can be detected in the receiver block, indicating that it could not be transported. This is polar transport. This indicates that auxin transport cannot be transported by diffusion or gravity. Some other mechanism must exist. Here is another example of this. Auxin can stimulate the formation of roots in woody cuttings. The transport of auxin stimulates adventitious root formation at the base of the woody cutting, whether they are placed upside down or right side up. The two on the left are upside down and the two on the right are right side up, thus showing polar transport from the top down to the bottom. So what is the mechanism of auxin transport? The chemiosmotic model of auxin transport explains how auxin moves directionally through plant tissues. This model relies on the pH difference between the cell wall and the cytoplasm, and the properties of the auxin's protonated and unprotonated forms. Essentially, auxin can enter cells passively via diffusion of its uncharged form, IAAH, and it is then trapped inside due to the higher pH in the cytoplasm, where it disassociates into its charged form, IAA-. This unidirectional movement is further facilitated by specialized efflux and influx carriers, creating a polar transport system. Shown here is one cell in a column of auxin transporting cells. IAA enters the cell either passively in the undisassociated form, IAAH, or by a symporter in the disassociated form, IAA-. The cell wall is maintained at an acidic pH by the activity of the plasma membrane ATPase, or proton pump. In the cytosol, which has a neutral pH, the anionic form predominates. The anions exit the cell via auxin anion efflux carriers that are concentrated at the basal ends of each cell in the longitudinal pathway. To make things more complicated, we now know that auxin is also transported non-polarly in the phloem. It can be used to transport small amounts of auxin out of mature leaves. It is the principal pathway for auxin transport in the roots. The auxin permease Auxin is expressed in a subset of columella, lateral root cap, and stellar tissues. It is asymmetrically localized on the plasma membrane at the upper end of the protophlom cells. These transporters become important in the root tip for auxin transport. This becomes important for gravitropic responses in roots. Molecular biology techniques give us the ability to label specific proteins so that we can visualize their location. Here in B, we can see the localization of AUX1 and auxin transporter as the fluorescent green color in the protophlom cells of the stele, a central cluster of cells in the columella and lateral root cap cells. And in C, LAX3, another auxin transporter mediates auxin uptake in epidermal cells surrounding emerging lateral roots to induce cell expansion and other changes that permit lateral root emergence. In this image, we can see the asymmetric localization of AUX1 at the base of each cell in the file of protophlom cells. There are other auxin transport proteins. Two types of transport proteins direct the flow of auxin throughout the plant: the PIN transporters and the ABCB transporters. Here we see in A that PIN proteins determine the basal direction of auxin movement. Directional auxin movement is associated with the tissue-specific distribution of PIN efflux carrier proteins. PIN1 mediates vertical transport of IAA from the shoot to the root in the apical meristem and in the root. AUX1 creates an auxin sink that drives basopedal auxin transport upwards from the root apex via PIN2 efflux carriers. Since some lateral diffusion of auxin may occur, pin 2 and pin 3 are thought to redirect auxin back into the vascular parenchymal tissue where polar transport takes place. This figure expands the view in the root tip that shows how the different pin transporters recirculate auxin in the root tip. The black arrows are pin 1, the brown arrows are pin 2, the red arrows represent pin 3, the green arrows pin 4, and the blue arrows are pin 7. Complicated. Auxin can also be transported by ATP-dependent ABCB transport proteins. The multidirectional arrows at the shoot and root apices indicate non-directional auxin transport. However, when combined with polarly localized pin proteins, directional transport occurs. ABCB4 regulates auxin levels in elongating root hairs. This image provides a model for how different auxin transporters function. The AUX-LAX transporters regulate auxin uptake into cells. The ABCB and PIN1-like PIN transporters control auxin efflux from the cell. The APB1 is an auxin-binding protein in the endoplasmic reticulum, and other ER proteins ILR1 and IAR3 deconjugate IAA. Other PIN proteins, such as PIN5, control transport across the ER membrane. So we can see that auxin transport is quite important and requires a very complex transport system. What do we know about the auxin signal transduction pathway? We know that tier 1 AFB proteins are the principal auxin receptors. We know that ABP1 functions as an auxin receptor that regulates trafficking of proteins. It's localized primarily on the endoplasmic reticulum. There is a small amount located at the plasma membrane, and if you knock out these genes, they are lethal to the plant. So they're necessary for a living plant. And it may stimulate proton pumping by increasing the retention of the H+ ATPases and auxin efflux proteins on the plasma membrane. Here is a model of how auxin activates transcription and gene expression. ARF is a transcription factor that, when bound by an AUX-IAA protein, forms a complex that represses ARF so that it cannot activate gene expression. In this figure, you can see a blue structural model of auxin, which can bind to its tier 1 ABF receptor. Once this has occurred, the auxin-bound receptor complex can bind to the AUX-IAA of the AUX-IAA-ARF complex, causing the AUX-IAA protein to fall off the complex and free up the ARF transcription factor. This allows the ARF transcription factor to bind with another free ARF transcription factor to form an ARF dimer. In this dimeric form, it can bind to the DNA and activate auxin-regulated gene expression. Meanwhile, the auxin receptor complex of auxin AUX-IAA protein and tier 1 ABF protein becomes ubiquitinated, which is the attachment of a small protein called ubiquitin. Ubiquitin acts as a tag and marks this protein complex, telling the cell that it should be degraded by the cell's protein degradation system, the proteasome. Auxin can induce the expression of well over 1,000 genes in the plant. Auxin induces genes and proteins that fall into two classes, early and late. Early genes and proteins, say within 2 to 60 minutes after exposure to auxin, are involved in signaling. Some of the proteins are already present to induce transcription. Some genes are involved in growth and development, such as AUX-IAA gene family, the SALR gene family, and the GH3 gene family. The early genes also include genes that are involved in stress responses, genes encoding the glutathione S transferases and the ACC synthase. ACC synthase is responsible for ethylene biosynthesis that I referred to before, which is another hormone. Late genes are involved in growth and development, and they're often induced by the early responsive transcription factors. They're required for long-term responses of auxin. Examples include auxin transporters and cell wall modifying proteins. Now that we know what auxin is and how it's regulated, let's discuss the multiple actions auxin performs in the plant. As we've seen at the beginning of this lecture, auxin can stimulate plant growth. This is a plot of a time course for auxin-induced growth of avena or oat coleoptile sections. Growth is plotted as the percent increase in length. Auxin was added at time 0. It is clear that IAA stimulates growth compared to the sucrose control. When sucrose, shown here as SUC, suc, is included in the medium with IAA, the response can continue for as long as 20 hours. Sucrose prolongs the growth response to auxin mainly by providing osmotically active solute that can be taken up for the maintenance of trigger pressure during cell elongation. Potassium chloride, or KCl, can substitute for sucrose. The inset shows a short-term time course plotted with an electronic transducer. In this graph, growth is plotted as the absolute length in millimeters versus time. The curve shows a lag time of about 15 minutes for auxin-stimulated growth to begin after administering a dose of auxin. This growth response is concentration dependent. In this figure, you can see a typical dose response curve for IAA-induced growth in P stems or oat coleoptile sections. Elongation growth of excised sections of coleoptiles or young stems is plotted versus the increasing concentrations of exogenous IAA. At higher concentrations above 10 to the minus 5 molar, that's 10 micromolar, IAA becomes less and less effective. Above 10 to the minus 4 molar or 100 micromolar, it becomes inhibitory, as shown by the fact that the curve falls below the dashed line, which represents growth in the absence of added IAA. As I mentioned earlier, this is due to auxin-inducing ethylene biosynthesis, a growth inhibitory hormone. So how does auxin stimulate growth? The acid growth hypothesis explains a lot of what is going on, but not all. The acid growth hypothesis states that auxin initiates an acidification mechanism resulting in a lowering of the pH in the apoplast. Enzymes involved in wall loosening are activated by the lowered pH, resulting in an increased growth rate. So what's the evidence for acid growth? First of all, we know that pH drops in the apoplast after addition of auxin. However, growth is stimulated for hours after addition of auxin. If you artificially lower the pH in the apoplast around a coleoptile, for example, it does stimulate growth by itself without auxin, but only for about 60 minutes. Furthermore, the lag time for this acid-induced growth is only 1 minute. As you recall, there was a 15-minute lag time for auxin-induced growth. You can also add neutral buffer to the medium and inhibit the auxin action by absorbing the protons and preventing the acid-induced growth. We also know that wall extensibility is increased by auxin, and we also know from what we've discussed earlier in our growth lecture, expansins loosen cell walls at acidic pH. They break the hydrogen bonds between the polysaccharides of the wall, loosening the cell wall and increasing the extensibility of the cell wall. This graph shows the kinetics of auxin-induced elongation and cell wall acidification in maize coleoptiles. The pH of the cell wall was measured with a pH microelectrode. Note the similar lag times, 10 to 15 minutes for both cell wall acidification and the increase in the rate of elongation. This increase in proton extrusion from the cells may result from the increased synthesis and/or the activation of the proton pumps. However, the acid growth hypothesis is not the complete answer. For example, Auxin alters transcription of many genes within 2 to 60 minutes after application. The acid growth is only sustained for 60 minutes, whereas Auxin-induced growth can be stimulated for up to 12 hours. There are other genes and enzymes that are likely to be involved in the sustained growth response that affect osmotic adjustment and polysaccharide synthesis as well. The current model for auxin-induced growth integrates auxin transport, gene expression, and cell wall mechanics. It involves the hormones' interactions with receptors, the regulation of gene expression, and subsequent effects on cell wall properties, ultimately leading to cell expansion and growth. This image is a simplified model for the IAA-induced proton extrusion and cell expansion from an annual review of plant biology article in 2020. Under low auxin conditions, on the left, basal plasma membrane, seen here as a PM, H plus ATPase activity is maintained through the balance of phosphorylation via an unknown protein kinase and dephosphorylation by pp2c.d protein phosphatases. In response to auxin, on the right, the Tier 1 AFB-mediated signaling promotes sour gene expression on the bottom left. Following protein synthesis, sour proteins directly interact with and inhibit plasma membrane localized pp2c.ds, preventing these phosphatases from dephosphorylating threonine 947 in the amino acid chain, and perhaps additional residues of the plasma membrane proton ATPase, thus keeping these proton pumps in the phosphorylated activated state. This increase in plasma membrane proton ATPase activity acidifies the apoplast, activating expansins, seen here as EXPs, and cell wall remodeling enzymes to loosen the cell wall. Proton pumping also hyperpolarizes the plasma membrane, which activates inward rectifying potassium channels and energizes proton-coupled anion symporters, seen here as X-. These transport activities maintain solute uptake needed for sustained water uptake and turgor pressure maintenance to drive cell expansion. So to summarize the general auxin effects on growth, auxin stimulates elongation growth in young stems and coleoptiles, and low levels are required for root elongation, but higher levels inhibit root growth by stimulating the production of ethylene. Phototropism is a kind of growth. It's a growth in response to light. This is a time course of illuminated and shaded sides of a coleoptile responding to a 30-second pulse of unidirectional blue light. You can see that the growth of the shaded side is stimulated by light, whereas the illuminated side is actually inhibited. There's evidence that the unidirectional light can stimulate lateral distribution of auxin in corn coleoptiles. The amount of auxin in the agar block is expressed as the angle of curvature the agar block induces when assayed by the coleoptile curvature bioassay. A shows the amount of IAA diffusing into the block in darkness. B shows the amount of IAA diffusing into the block in unilateral light. C shows the lack of light-induced IAA migration to the shaded side in a coleoptile tip divided by a mica barrier. And D shows the light-induced migration of IAA to the shaded side in an intact tip with auxin amounts much higher than the illuminated side. Newer molecular biology techniques confirm these results. Here we see the lateral redistribution of auxin during phototropism, which is visualized by transforming plants with a DR5 Gus reporter gene construct. The construct is a sensitive reporter of the presence of auxin. On the left, you can see the lateral auxin gradients are formed in Arabidopsis hypocotyls during the bending response to unidirectional light. Auxin accumulation on the shaded side of the hypocotyls is indicated by the blue staining. On the right, you can see the treatment with the auxin efflux inhibitor, NPA, blocks both phototropic bending and auxin redistribution. A similar redistribution of auxin occurs during gravitropism, which brings us to gravitropism, which is a growth in response to gravity. One can have negative gravitropism, where shoots grow in opposition to the gravitational force, and positive gravitropism, where roots grow in the direction of the gravitational force, as seen here. In negative gravitropism in a shoot, auxin is transported to the lower side of a horizontally oriented oat coleoptile tip. One piece of evidence for this is shown in this figure. In A, auxin from the upper and lower halves of a horizontal tip is allowed to diffuse into two agar blocks. In B, the agar block from the lower half, the left, induces greater curvature in a decapitated coleoptile than the agar block from the upper half on the right. Roots are a little more complicated, I'm sorry to say. The perception of gravity in roots occurs in statocytes in the center of the root cap. Statocytes are specialized plant cells that play a crucial role in gravity perception. These cells contain dense, starch-filled organelles called statoliths, which settle to the bottom of the cell due to gravity. This sedimentation process triggers a signaling cascade that ultimately leads to the plant reorienting its growth in response to gravity. In A, you can see an electron micrograph of a root tip showing the apical meristem, labeled M, columella, labeled C, and peripheral cells, labeled P. Figure B is an enlarged view of a columella cell, showing the amyloplasts resting on top of the endoplasmic reticulum at the bottom of the cell. Amyloplasts are starch-containing plastids. In figure C, you can see a diagram of the changes that occur during reorientation from the vertical to the horizontal position. Part of the signal transduction pathway involves changes in the cytoplasmic pH of the columella cells. A pH-sensitive dye can be used to illustrate this. In A, we see a micrograph showing a magnification of the root tip and two columella cells at different levels, or stories, of the root cap, labeled S2, story 2, and S3, story 3 in the inserts. The cytoplasm of the two columella cells are fluorescing because the cells have been micro-injected with a pH-sensitive fluorescent dye. The vacuoles, V, contain no dye and therefore appear dark. In B, we can see how the cytoplasmic pH increases in less than 1 minute after gravostimulation. In C, we can see the actual imaging of the pH-sensitive dyes and the response of the two columella cells to gravitropic stimulus. The color scale below is used to generate the data in B. The root cap is a source of an inhibitor to growth, as seen here in this figure. If one carefully removes the cap, the growth of the root is stimulated. However, if one removes only half of the cap, the growth causes bending towards the half that has the remaining half cap. Likewise, if one places the intact root into a horizontal position, the root will grow positively in the direction of gravity. However, if the root cap is removed, the root will grow horizontally, not perceiving the gravitational force. Here we can see the current model in 2010 for root gravitropism. This model is still valid today. In the vertical orientation, IAA is synthesized in the shoot and transported to the root in the stele. In the root tip, the statoliths in the cap settle to the basal ends of the cells. Auxin is transported equally to all sides. Once it reaches the tip in the stele, it is then transported back up, basipedally, through the root, through the outer cortical cells, where it regulates the growth of the cells in the elongation zone. When a root is in a horizontal orientation, a transient lateral auxin gradient is established, primarily through the redirection of auxin flux from both sides of the root apex to the lower side. This redistribution is triggered by a gravity stimulus and is sufficient to induce changes in auxin-responsive gene expression and asymmetric root growth. This transient auxin gradient is sufficient to induce changes in the expression of auxin-responsive genes like IAA1, IAA2, and ARF19. The elevated auxin levels on the lower side of the root then inhibit cell elongation, causing the root to bend downwards in response to gravity. One way this model has been supported can be seen here, where a fluorescent auxin reporter gene was used to determine the auxin distribution in a root tip, in the vertical position seen in B, and in the horizontal position seen in C. You can see the green fluorescent color in the lower side of the root after moving the root into the horizontal position. Another important action of auxin is called apical dominance. The growth of axillary buds are inhibited by auxin in the intact plant with an active shoot apical meristem. Removal of the apex, or apical meristem, releases the axillary buds from apical dominance and allows them to grow. I did this cool experiment in my very first plant physiology class as a student. One can apply IAA in a lanolin paste in a capsule and place it on the stem where the apical meristem has been removed. The auxin in the capsule mimics the auxin in the apical meristem and prevents the outgrowth of the axillary buds. I thought this was a really cool experiment, and one of the reasons I started to get interested in plant physiology. Auxin is also involved in fruit development. The strawberry fruit is actually a swollen receptacle whose growth is regulated by auxin produced by the seeds, which are actually achenes, the true fruits. Receptacles are technically the fleshy base of a flower. The achenes are very small fruits with one seed inside. When the achenes are removed, the receptacle fails to develop normally. Spraying the acheneless receptacle with IAA restores normal growth and development, so auxin can regulate fruit size and shape. Another cool action of auxin involves tissue repair of the vasculature. In this example, the stem of a cucumber plant was decapitated and the leaves and buds above a wound site were removed to lower the endogenous auxin. The wound was a partial nick in the stem, enough to cut through the vascular strands on one side of the stem. Immediately after the wounding, IAA in a lanolin paste was applied to the stem above the wound. After about a week, you can see that xylem differentiation occurs around the wound following the path of auxin diffusion. This action is in part what allows one to graft a shoot onto a root and make a whole plant again. Wow, isn't auxin an interesting hormone? In this lecture, we talked about some of the important actions of auxin, particularly growth and development. I explained how auxin concentrations are regulated, how it transmits signals altering the pH, ion transport, water uptake, and biochemistry of elongating cells, and how it alters gene expression. Stay tuned for future videos on plant physiology, on grape growing, and on winemaking practices by subscribing to my YouTube channel. And if you like this video, then please like it on my YouTube channel so it brings to attention to others about this video. Have a great day.